sequencing is a technique used to determine the order of DNA nucleotides within a strand of DNA. A single-stranded piece of DNA is used as the template strand to carry out the reaction. First, a primer sequence is added to the DNA, and then the DNA is amplified by PCR. A primer is added to the amplified DNA that binds to the complementary sequence on the template DNA and instructs DNA polymerase where to begin copying the DNA. The key to the sequencing reaction is the addition of fluorescently labeled nucleotides to a much larger mix of unlabeled nucleotides. The labeled A, T, G, and C nucleotides each have their own label color. These nucleotides are chemically modified so that they can be added to a growing DNA strand, but no other nucleotides can be added to the strand after them. When the DNA sequencing reaction begins, the primer binds to the complementary sequence on the template DNA strand. The polymerase then begins to copy the template DNA by adding nucleotides to the growing strand. Most of the nucleotides that are added to the growing strand are unlabeled, but eventually one of the labeled nucleotides will be added. And because other nucleotides are unable to bind to the fluorescently labeled nucleotides, this causes chain elongation to stop. The new strand of DNA is detached from the template, and the process is repeated over and over again until many DNA fragments of varying lengths are created. The reaction is repeated enough times so that there will be strands of DNA that incorporated a fluorescent nucleotide at each nucleotide along the template DNA. Finally, the DNA fragments are rearranged in size order by running the sample through an electrophoresis gel, which separates them according to length, and they are passed in front of a laser that can read the identity of the fluorescent nucleotide blocking the end of each new strand. The sequence of labeled nucleotides is complementary to the sequence of the original strand. DNA sequencing techniques are used to determine the identity of nucleotides in short template DNA strands, but the same techniques can also be used to sequence the entire genome of an organism. The genome is copied multiple times, and those copies are broken into millions of pieces using restriction enzymes. Computer software is then used to align all of the small sequence fragments. When sequences of fragments overlap, they are merged to form one larger sequence. When all of the sequence fragments are put back together, the sequence of nucleotides for the entire